Hello and welcome to Nicholas Genetics Lessons. In this video I'm going to explain what is a transformation is, and we're going to solve a problem. And here's a problem. Frederick Griffiths accidentally discovered transformation when he attempted to develop a vaccine for pneumonia. He injected mice with samples from S strain virulent and or R strain non-virulent pneumococci bacteria, Streptococcus pneumoniae. Which of the following results is not consistent with Griffith's experiments? And I want to underline the word not. So let's check our answers. And by the way, you can stop video here. You may also try to choose which answer is correct or non-correct, and when you would be ready you can run video again and you can compare your answer with my answer and explanation. So answer A, injected S strain mouse dies. And this is correct answer because S strain is virulent. So of course if we would inject a mouse with this strain, this would die. So this answer is correct. And next statement, B, injected R strain mouse lives. And as you see from our problem, R strain non-virulent. So this means that if we inject mouse with this strain, this would live. And this is also correct answer. Next statement, C, injected heat killed S strain mouse lives. And of course if we kill virulent strain and inject a mice would also live because this bacteria wouldn't be able to reproduce, it would be killed. So this is correct statement. And next statement, D, injected mixture of heat killed S strain and live R strain mouse lives. And this is not true because those here we have one strain that is doesn't cause harm to the mice, R strain, and it is live. And we have also S strain that also when it's heat killed wouldn't do harm to the mice. Actually what happens inside the mice when we have for example, this is going to be a cell of the bacteria, and within a blood vessels of the mice it can reproduce, and this is going to be R strain that is not virulent, but also these mice were injected with heat killed S strain, and that means that this bacteria would be degraded within body of the mice, and its DNA would be fragmented to small pieces. And when time passes, these fragments would be even degraded to smaller and smaller pieces. And when we have a mixture of bacteria that is not virulent and it has its own DNA inside, and as you see, some of these pieces from the virulent bacteria can have one fragment that codes for the gene that cause virulence in the S strain. So if such piece would be acquired by the bacteria, and bacteria needs some building materials from its surrounding environment, such a piece of the DNA, it's not necessarily have to be the whole gene. It can be only a fragment of the gene that is different, because probably these two strains of the R and S strain bacteria would have all the same genes, but one gene would cause virulence in one strain of the bacteria. Probably it has just few mutations, and if such a fragment would be acquired by the bacteria within a cell, it can find its homologous piece on the DNA of the bacteria, and such two pieces can be exchanged. So here is another picture of this same process on the bigger scale. So this is DNA of the bacteria, and here is a fragment on this piece of the DNA that codes for the certain gene that cause virulence or not virulence in bacteria, and here is the same fragment from the virulent strain of bacteria, and they would exchange. And why this happens? Because this is normal process of the DNA repair when it's break within bacteria. So when it's break, certain proteins or enzymes would do a repair, and sometimes they wouldn't understand that this piece or fragment of the external DNA don't belong to this bacteria, and they just exchange these two fragments, and external fragment becomes a part of the bacterial DNA. And now the last statement E, injected mixture of heat-killed R strain, and we know that R strain is non-virulent. So if we heat kill it, it doesn't affect if mice would live or not, because anyway this is harmless strain. And live S strain, that virulent, if it would be injected in mixture, of course mouse will die. So this is true statement. So the only statement that contradicts with Frederick Griffith's experiments would be answer D. And I also want to add that transformation is not the only way how bacterial cell can get foreign DNA. The other two variants would be conjugation, when two cells of bacteria would come into the contact and would exchange DNA, and the third variant would be transduction, when bacterial cell would be infected with bacteriophage, and bacteriophage can also move some pieces of the bacterial DNA between different strains, and also this is the way how some bacteria can get foreign DNA along with bacteriophage DNA. And my last comment would be, we don't use this term transformation when we talk about eukaryotic cells, cells of the animals or plants, because transformation is reserved in order to describe different process when normal cells become cancerous. So in order to describe the same process in eukaryotic cells, we use different term. So instead of transformation, we call such process transfection. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. And see you in the next video. Goodbye.